And so are we live now, Anna?
Yeah, now we're live. Yeah.
Fantastic. All right, well, let's go ahead and get started. We have today Srinivas Niranj Chandrasekaran. I'm not sure I pronounced that correctly, Niranj. But as a brief introduction for Niranj, he obtained his PhD in Charles Carter's lab at UNC at Chapel Hill. And after completing his thesis on statistical mechanics and protein dynamics, Niranj moved to develop his interest more in bioinformatics and integrative biology. He's currently completing postdoctoral training with Ann Carpenter's lab at the Broad Institute in Harvard. And there he's leading a collaboration of 13 pharma companies and nonprofit organizations to generate the largest publicly available cell painting data set. And he's going to talk about what cell painting is and his research program there. In essence, he's developing machine learning-based analysis pipelines with the goal of accelerating drug discovery, and is very relevant for our Illuminating the Druggable Genome mission. He recently published an incisive review in Nature Reviews Drug Discovery. If you are interested in the topic and you want to read more, it's focused on image-based profiling for drug discovery. I think it's fair to say that despite his junior status, he's already beginning to establish himself as a leader in this field. So please welcome me, please join me in welcoming Niranj. Thank you very much for sharing your work, Niranj. I'll turn the stage over to you.
Thanks for the introduction, Michael. I'll quickly share my screen. All right, I hope everyone can see my screen. So yeah, good morning, everyone. I hope everyone's having a great day today. As Michael said, I'm Niranj Chandrasekaran. I am a postdoc in the Carpenter Lab at the Broad Institute. It's also called the Imaging Platform. And thank you so much for the invitation. I'm quite excited to share the work of the Imaging Platform with all of you today. Today, I will be introducing you to the field of image-based profiling by giving you a general overview of all the work that's happening in the Imaging Platform and how that's transforming the drug discovery process. It's a relatively new field and has quite a lot of potential. And towards the end of my presentation, I will talk to you about this exciting project that I'm currently working on, the one that Michael had already introduced to you. So the way I've structured the talk today is this. I will be trying to answer 4 main questions regarding the use of cell images in the drug discovery process. The questions briefly are, why use cell images? What kind of information can you extract from cell images? What can this information be used for? And then finally, if someone is interested in this information, where can they find that? So let's get into the first question. So what makes the cell images special for answering biological questions? Before I answer the question, I just wanted to show you this set of stunning images taken by Derek Sung at the University of Pennsylvania. And while you're looking at this, I will tell you, begin to tell you some of the advantages of using images for the process of, for identifying or for characterizing biological systems. One main advantage of using images is that not only do these images look very pretty, but there is a lot of information that you could extract from them. Given that the 4 images that you see here, the cells look completely different from each other, and there is a lot of information that could be useful for the drug discovery process. I'll get into that a little bit later. The other advantage of using images of cells is that by definition, it offers you a single cell resolution. So a recent trend in biology, especially in the field of transcriptomics, is a pivot towards using single cell technologies in the last few years. But microscopy has been a single cell method ever since the microscope was discovered 400 years ago. So that is an additional advantage of using cell images. The most important advantage, what I consider to be the most important one, is that using images to answer biological questions or doing image analysis is very quantitative. So here is an example where if we were looking for a drug that could induce cells to go into a polyploid state. So the one on the left is not, the one on the right is. And this is a useful characteristic in cancer treatment. Then one could just measure the total number of total amount of DNA inside the cells, and then look for compounds that is able to do this. So this is an approach that one of our collaborators took, where they took a certain form of leukemic cells, and then treated them with several drug molecules, and they were able to identify potential drugs that were able to increase the DNA content. So that example provides us with a great segue into our next question, which is, if image analysis is quantitative, how can one extract information from cell images? Traditionally, microscope experiments have focused on measuring one or two things of interest from images. Like I showed you in the previous slide, where we were looking for the total DNA content of the cell. Here is another example, a work by one of our collaborators, where they were trying to distinguish leukemic cells from healthy cells. This was, distinguishing these two is crucial for the treatment of leukemia, but the problem here was that identifying the difference between them was not that easy. But when they looked at the images of these cells, what the collaborators identified was that the leukemic cells had this tendency to form the cobblestone-like pattern that you see on the left. And what they saw was that the healthy cells did not have this behavior. So now the quest became to look for compounds that would be able to stop the cells from forming these clumps, these cobblestone-like patterns or form groups which look like these cobblestone patterns here. So these kinds of experiments, the one on this slide and the one on the previous slide, where we're looking for one or two measurements or one or two characteristics of the cell at a time, is generally what is called a screening experiment. I would now like to contrast this with a different kind of experiment, still with images, called a profiling experiment. So about a decade ago, the trend started changing slowly in the field of microscopy, especially in the Carpenter lab, where we began measuring not just one or two features of the cell, but rather we started measuring as many things as we could. This gave rise to the motto of the lab, which is measure everything and then ask questions later. So you measure everything, whatever you can, and then later decide what to do with those measurements. Measuring just one or two features like we do in the screening experiments is like having a huge data set focused on, and then focusing just on one column of data and then just getting rid of everything else. This is definitely, it's a shame if you're throwing away so much biological information, because cells are rich in information content. So we now encourage everyone to perform profiling experiments where you extract as much as possible and in a non-biased manner, and then worry about what to do with them later. So the rest of my talk is going to focus primarily on profiling experiments and their applications. So there are several ways in which one could perform a profiling experiment. The way we do that is using a specific assay in our lab, developed in our lab, called the cell painting assay. This was actually developed in collaboration with the Stuart Schreiber lab, also at the Broad. What we do in this experiment is that there are 6 different dyes that label 8 different cellular components. And then once the cells have been stained, then they're imaged in 5 channels or wavelengths that are shown in this figure here. This assay is quite inexpensive, and it's also a non-fussy assay. And it makes it, and these characteristics make this assay amenable for use in high throughput experiments. So all the results that you see that I'm going to show you today are based on the cell painting experiment. So once you have the cells stained and they have been imaged, the next thing that we do in our experiments is to extract features, extract information from these images. Typically, we measure some simple things, such as the counts, the number of cells that you have in your image, and other counts that are relevant, the sizes of the different compartments of the cell and the cell itself, or the fluorescence intensities of the dyes. So these are some of the simple features, but there are other complex features which are also measured, Some of them are inherently non-parametric, such as the shape and the texture of the cells. We also measure correlations between the different channels in which we image the cells, and also measure other things such as the neighborhood relationship between the cells. So in the end, we end up with thousands of measurements per every single cell. So the next question that may have struck you is that how do you make these measurements themselves? For this, we use our open source image analysis software called Cell Profiler. It's a widely used software, not just in academia, but also in pharmaceutical industry. And if you're more interested in knowing or using this software, you could check out cellprofiler.org. So from Cell Profiler, what we get is a bunch of hand-engineered features. So these are features that we know either have mathematical or biological meaning, and we know them beforehand before we even try to extract those features from the cells. In recent years, there has been advancement in a new type of feature extraction, which is based on deep learning and computer vision. And these are the next generation computational techniques and image analysis methods. It would already be quite obvious to you, or you may be aware that this is something that's possible, where the same architectures that you use in order to find faces in the images on the left can actually be trained to identify nuclei on the right. All that you need to do is to provide the system with sufficient training data. So this is an exciting field of research that's happening currently in this space, and there'll likely be a lot of interesting papers that will probably come out in the next few years. So once the features have been extracted from the cells, the next step in the experimental pipeline is to make use of these features in order to make biological predictions. So that's where the profiling experiment comes into the picture. So the basic idea behind every profiling experiment is quite simple. What we do is we take cells, perturb them with several different perturbations. And these perturbations could be simple, just adding compounds to the cells, or it could be more complex, such as having genetic perturbations, where you're either overexpressing certain genes in the cell, or you're knocking out certain genes in the cell. And then what we do is perform the cell painting experiment, extract the features, and we get a list of features for every single cell in the subject to that particular perturbation, and then finally do some sort of an analysis with those profiles. And a very typical profiling experiment could be as simple as just finding similarities and differences between the profiles. And there are several different ways of doing it. In the slide I've shown here, we do a simple clustering, which puts all the profiles that look like each other together and the ones which are dissimilar far away from each other. So that was a very brief introduction to what a profiling experiment actually is. And now I will show you the power of this profiling experiment and this profiling approach with the help of some examples of image-based profiling and how it has been applied in order to accelerate drug discovery. There are several applications of image-based profiling in the context of drug discovery, but today I'm going to focus only on two applications given the time constraints. The two different applications that I'm going to focus on are, well, one of them is in the compound perturbation experiment, where we are identifying the mechanism of action of a compound. And the other is a genetic perturbation experiment where we are identifying the functions of a gene. I'll go through each one of them separately now. So let's take a look at the compound perturbation experiment. The idea behind this experiment is quite simple. You take a particular compound of interest, you perturb cells with those compounds, and then do cell painting analysis and measure its features and profile. Then what you do is to compare it with a library of other chemicals or genetic perturbations that you may already have. And then the MOA, or the mechanism of action of this compound, is assigned to the compound by a guilt by association approach, which is that compounds that have similar profiles likely have similar MOA. This is a very simplistic approach to assigning MOAs, but it is quite successful for many compounds. And I'll show you examples of that in the next 2 slides. So by using these approaches, one could quickly learn the compound's MOA without performing much more complex assays in order to ascertain what the mechanism of action of a particular compound would be. And thus it helps in the drug discovery process. So the image-based profiling can serve as a primary screening method in any drug discovery pipeline. So let's look at an actual example of such a compound perturbation experiment. This is an actual experiment over a decade ago, but it's still quite relevant. And this is one of the most common applications of image-based profiling in both academia and in the industry. So in this plot here, you can see that the predicted and the actual MOAs of the compounds are plotted. And the numbers in these squares tell you the number of compounds that belong to each MOA class. And from this image, it might be clear to you, or it might be obvious to you, that the predicted class and the actual class seem to match quite a lot for most of the compounds in this experiment. And it is also nice to see that compounds which have the same MOA, they seem to cluster together, given the high numbers of the values that you see along the diagonal. But this experiment was a very simple case, and we were using compounds which had very strong phenotypes and which were very obvious. So it wasn't very difficult to perform this experiment or to generate these results. It was quite expected. So recently, a couple of our collaborators have been working on a more complex experiment where they screened over 1,000 compounds in a similar fashion to the one that I showed you in on the last slide. And here you can see that there is some pattern along the diagonal, but in general, we don't see that. And this is not unexpected because many of these compounds had overlapping MOAs, so they aren't expected to cluster along the diagonal like they did in the last experiment. But it's still very encouraging to see that there is some pattern along the diagonal. What was interesting about this experiment was that apart from having image-based profiling data for this set of compounds, we also had transcriptional profiling data, transcriptional profiling in specifically from a high throughput transcriptional assay, L1000. And here you can once again see the same plot where we are trying to assign, see if the predicted MOA and the actual MOA match. You can see that the information in this plot is slightly lesser than what you had in the last one. But what was interesting to us was that for many compounds, we found the data in the morphology space to be quite complementary to what was there in the transcriptional or the transcriptomics phase. So the idea here is that we are very encouraged to make use of profiles from both morphological profiling and transcriptomic profiling and merge them together into a joint profile and then make predictions of the MOA of the compounds. And this is an effort that is actively ongoing, and we believe that this will increase the power of the profiling approach itself. Now, let me show you the other example from a genetic perturbation experiment. And the approach here is quite similar. What we do is we take a single, or we take cells, we perturb them, or we perturb the genes in some form, either by overexpressing the genes or by knocking them out, and then generate the profiles and then compare them against a reference library of compounds or genetic perturbations. And from this, we assign the functions of a gene by using a similar guild by association methodology. So let's look at a result from an actual experiment. Here, we tested a bunch of genetic perturbations. We overexpressed a number of genes, and we were very happy to see the clustering in the data. And it was quite similar to what we saw in the compound experiments as well. And so in this experiment, we overexpress the genes, cell paint them, and then measure the features and then do a clustering. And what we find is that we rediscover a lot of biological pathways. For example, the RAS pathway is up here, and then the Hippo pathway is up down here. And what we find is that the several genes that are part of these different pathways seem to cluster together by just using their morphological profiles. This kind of a methodology can be used for identifying gene functions or the gene pathways of certain genes, but it can also be used for stratifying patients and characterizing disease alleles that contribute to different morphological profiles. So here is an example from lung cancer. Like with most diseases, it's not a single disease, but instead can be caused by a variety of underlying mutations. So what we do here is that we overexpress these different alleles of this of several genes that are associated with lung cancer. And what we, in this example, there are 353 of those different alleles. And what we find is that once we have the profiles and then plot them in this heat map, we find that it's not that there are 353 different types of cancer, but rather a bunch of fewer subtypes, but each of these subtypes have a number of different alleles that cause that particular subtype. And this result becomes much more obvious when we're looking at a specific example of a specific gene. For example, in this, we have the BRAF gene here. We overexpress this gene along with a bunch of number of other alleles of this gene. And then what we find is that there is a group of these alleles on the right top corner, which seem to be quite distinct from the wild type at the bottom here, given the color of this the heat map. It's quite blue, which means that it's quite different from the wild type. And this was something that was already known and had been well characterized. So it was very good to rediscover or at least replicate what is already known. And we also find a bunch of other alleles which seem to be completely different from the wild type and this particular group at the top. So there are several other applications of image-based profiling. And as I said, I wasn't able to go into the details of all of them, but I have linked 2 different review papers here, which go into the details of these applications in more detail. So now we have seen how image-based profiling experiments are performed and what sort of applications or what sort of things that we can use image-based profiling for, especially in the context of drug discovery. At this point, you may be curious to know if there is any data sets or reference data sets that you could use, or if you perform your own profiling experiment, would you be able to query a database and compare your profiles with already existing compounds? So this is where I want to bring your attention to this new project that I am currently working on. This project is called Jump Cell Painting. And I am currently working on this, and this is an ongoing project, and we'll probably have more information from this project in about 18 months or so. So JUMP stands for Joint Undertaking in Morphological Profiling. 10 pharmaceutical companies and 2 nonprofits got interested in using cell painting data and image-based profiling as a part of the drug discovery pipeline. So they got together and formed this consortium to create the largest publicly available cell painting data set. The data set that we generate will have data from 140,000 compound perturbations and 10,000 genetic perturbations. And all these experiments will be performed in 5 replicates across multiple different locations at different pharma companies and nonprofits. And through this data generation process, we aim to address several assay development and computational challenges in cell painting that could further accelerate the use of cell painting in drug discovery. We will generate not just profiles, classical profiles, hand-engineered profiles from Cell Profiler, but we'll also be generating deep learning features. And this, along with images, raw images of over 1 billion cells, will be released to the public. So when I spoke to Dr. McManus a month ago, he had suggested that I take a look at the number of genes that are part of the jump cell painting project and the list of genes that the IDG is interested in. At the moment, it looks like there are 155 genes in the IDG list that are also common to the list that we have, we are going to profile during jump cell painting. And we may actually increase the number of genes that we are profiling, so the OLAP could actually increase. So in case you are interested in a particular gene and you're looking for drugs that may target that particular gene, you could always query the data set that we are going to generate, or you could generate these data sets on your own and then query them to the compound library that we are also going to generate. And then perhaps you might find some matches or anti-matches, and that might give you some candidates that could actually, candidate drugs that could actually be targeting your gene of interest. So I really hope that this slide piques your interest in this project. There are a few frequently asked questions about this project. When will the data be released? Likely on November 1st, 2022. We have had some COVID delays, so we have had to move the dates quite a bit. But at this point, it looks like it will be November 1st, 2022. The data production will likely begin next month. And what we are doing now is we are optimizing the different experimental conditions, all the experimental stuff that I spoke about in relation to cell painting, and also developing computational methods that will be used for analysis. And the last question is, can you join the consortium if you're quite interested in this? Well, it's a little too late to join as a data production partner, but we are always open for collaborations. We have multiple collaborators already working with us using our data to do their analysis. And if others are interested in it, we definitely welcome collaborators. So that was my last slide. I'm very grateful to everyone on this slide for generously sharing their data and figures with me so that I could share them with you. And you can get in touch with me via email or Twitter that I've linked here. And if you have any questions that I'm not able to answer today. And thank you so much for spending your time with me. And I'll be happy to take your questions now.
Great. Thanks very much, Niranj. I'll begin. There is one question in the chat box. Can you see this question from Mark Haynes?
Yeah, let me open it up. All right. So yeah, so I'll just read out the question. What would be the average time for assay development to library screening of 10^5 compounds? Can this technology be used in 1536 well? And what is the time to collect one plate of raw data? Yeah, so the first question is that the data production for the 100,000 compounds or so, it's going to take us approximately 4 months. That's what we assume. At the Broad, it happens a little bit faster because we have been working on this technology for a long time. But for anyone who is new to this field, it takes some time for them to get used to it. So we assume that they'll take some more time, but we assume it's between 4 and 5 months. The second question, can this be used on 1536 well plates? Yes, some of the partners in this project will actually be generating their data in 1536 well plate, but in general, we use 384 well. But 1536 also works. The main issue with 1536 is that we don't have enough fields of view because of the size of the wells. So you can't take enough images per well, but we find that that's not a major issue. And the time to collect one plate of raw data, I'm not entirely sure, given that I'm not the experimentalist, I do the computational work, but based on my discussion with people, the experimental people, I think that the image acquisition takes about 90 minutes per plate. And yeah, the data, the sample preparation stuff will, of course, add more time to that.
Thank you. And I'll note that Niranj's email is down below. It's for anyone that has any follow-up questions. Of course, you can feel free to add it in the chat, but don't hesitate to reach Niranj by email. For the sake of time, let's move on to the next presentation from Stefan Feske. Some of you may know Stefan through the IDG program, where he explored an understudied calcium channel subunit in T cells and its relationship to immune responses. So Stefan's a professor and vice chair in pathology. He's also the Jeffrey Bergstein Professor in Medicine at NYU. He graduated summa cum laude with the MD from the University of Freiburg following a residency in rheumatology, and then did his postdoc at Harvard Med School. He joined NYU in 2007. And Stefan's lab is basically investigating how ion channels regulate immune responses in human and mice. Without undue delay, I'll just please join me in welcoming Stefan. It's really great to have him here. He's been a leader in this field for nearly 2 decades. It's really a privilege to have him join us. So please, Stefan.
Thank you, Michael. Thanks for the generous introduction. Let me share my slides. Let's see. Okay, can you see my slides?
Yes, we see them.
Perfect, great. So this will be a real change in topic following on the previous really beautiful talk. So I will speak about our attempts to identify new ion channels and transporters in immune cells. And this is a fairly new development for us in the last couple of years that we have started. My lab has traditionally studied the role of crack channels, calcium release activated calcium channels, the main calcium import channel in immune cells, or in T cells at least. And we have since then expanded our efforts to other ion channels. And the reason why we are doing this is because we feel that ion channels are not well studied in the immune system. There are some labs who study them, but compared to neurons and cardiomyocytes, kidney, and some other excitable organs and cell types, ion channels in the immune system are not really well studied, which has to do with the fact that immunologists are not really familiar with the methods used here and maybe the background of ion channel physiology, and vice versa. Immunology is a daunting field if you're not a trained immunologist. So, and I will describe today our efforts to identify ion channels that regulate immune function. So because most of you will not be immunologists, I just want to very briefly show you the 2 main parts of the immune system, which we typically divide, which is an oversimplification into innate and adaptive immune, the innate and adaptive immune system. The innate immune system consists of these cells here, mostly macrophages, neutrophils, mast cells if you have allergies, dendritic cells. And what these cells do is they can recognize pathogens like bacteria, viruses, based on surface molecules that are specific to these pathogens, but that don't occur in mammalian cells. And they can do this very quickly. On the other side of the equation are adaptive immune cells, T cells and B cells mostly, that respond much more slowly, but they have the advantage that they can be much more specific because they can rearrange and optimize their cell surface receptor, the B cell receptor and the T cell receptor, to achieve highly specific responses. And they also can develop a memory. They can basically remember whether they have seen a pathogen before or not. And there have been maybe a dozen or maybe 10 to 20 or so ion channels that have been described in the context of the function of immune cells. And this slide provides an overview. This is a virtual immune cell, not any particular immune cell, and illustrating some of the ion channels that have been implicated in immune function. As I said, my lab is mostly studying, or has mostly studied, the Ori channels, which form the pore of the crack channel, the main calcium influx pathway in T cells. There are P2X receptors that are gated by ATP that also mediate calcium influx or the efflux of potassium. There's TRPM7, the magnesium channel that's important for T cell development and other immune functions. There are potassium channels like KB1.3 and KCA3.1 that set the membrane potential and provide the electrical driving force for calcium and other ions into the cells, and a number of other ion channels that have been studied in patients and mouse models. But overall, there's relatively little information about ion channels in immune cells. And this is in particular true when you think about the entire universe of ion channels. There are several hundred ion channels and transporters annotated on the IRFA website. And this is illustrated by this cartoon here. You have trip channels, potassium channels, voltage-gated sodium and calcium channels, and so forth. So what we have done in the last couple of years is we have generated our own database that basically compiles these channels and transporters, plus additional proteins that regulate them, and newer channels into our own database, which we call the ICT library, Ion Channels and Ion Transporter Library. And it contains ion channels, transporters, regulators, and some proteins with enzymatic function. And so overall, our library contains 763 genes that we are testing in our systems. We also have a library of metabolic transporters, but I'm not really going to go into that today. So we are using two approaches to study the role of ion channels in the immune system. One is an expression analysis by querying this library across all sorts of immune cell subpopulations. And we're also doing forward genetic screens by shRNA and CRISPR. And I will give you examples of these methods in my talk. So the first analysis that I'm showing you is a principal component analysis of these 763 ICTs, ion channels and transporters across more than 100 immune cells and their gene expression based on RNA-seq data from the so-called IMGen database. This is a curated database to which multiple labs are contributing expression data. And what this PCA analysis is showing you is that B cells cluster very differently from CD4 and CD8 T cells or natural killer cells, macrophages and dendritic cells. And what this tells you is that we can identify different subsets of immune cells purely based on their expression of ion channels. And presumably, different ion channels regulate the function of these immune cells. We can show this in more granular detail by looking at a heat map in which each column is a cell type and a subset of these cells, and each row is an ion channel. And what you can see is that, for instance, B cells express a very specific cluster of ion channels that is different from ion channels expressed in dendritic cells or neutrophils down here. My lab has been mostly interested in T cells in the context of Ori and crack channels. So we looked in T cells first in a little bit more detail. And what we find is that of all the ion channels that are in our database, there are several of those that have been described to play a role in T cell function are also fairly highly expressed in T cells. So they're indicated by the yellow bars here. But there are also other ion channels that are highly expressed in T cells for which a function in T cells or immune cells has not been described. So we think there is really a potential here of describing more ion channels that are expressed in T cells. And I want to give you two examples how this expression analysis may actually identify these channels. One is LRRC8C, which is a chloride channel. I will come back to that. And the other one is a subunit of the sodium-potassium ATPase that's fairly selectively expressed in T cells. And I contrast the expression of these channels in T cells to that of ORI1, which is really important for T cell function, but is neither highly expressed nor selectively expressed. So we think that this expression analysis has some value. So the first example is this subunit of a sodium-potassium ATPase, which is enriched in T cells, but not so much in other immune cells. This is a project driven by Axel in my lab, a postdoc, who has been at the forefront of this project for a long time. And when we look in other tissues and compare the expression of this subunit, which I call C here, to the expression in other cell types outside the immune system or other organs outside the immune system, we find that this subunit is when we delete it by shRNA, we find that we can basically suppress the proliferation or the expansion of T cells in vitro. This is after stimulation with T cell receptor antibodies in vitro. And the reason for that is that T cells do not proliferate. This is measured by CFSE, which is basically a cell dye that is taken up into cells and is diluted with every cell division. And the less signal you have, the more the cells have proliferated. And you can see that the cells in which this subunit is missing, they don't divide as much. And we can also test the role of this ATPase subunit in vivo by using a disease model that we often use in this context, which is the experimental autoimmune encephalomyelitis model. This is a mouse model of a human disease called multiple sclerosis, which is an autoimmune disease of the brain and the CNS, in which autoreactive T cells that make cytokines like GM-CSF and interferon gamma invade the brain and recruit other immune cells and eventually lead to the destruction of the myelin sheath surrounding myelinated neurons. These are so-called Th1 and Th17 cells. And what we can do is we can take T cells from transgenic mice that have a T cell receptor that specifically recognizes myelin, transduce them with the shRNA against the subunit, and then take these T cells and inject them into mice to induce EAE, and then we can look how that affects disease development. You can see that when we delete this ATPase subunit, we can basically prevent EAE compared to the control mice. And the reason for that is that we do not get an accumulation of T cells in the spinal cord, in the CNS of these mice compared to the controls. This is quantified here on the bottom right. And this is specific to the spinal cord, and because we see similar numbers of T cells, control and shRNA treated cells in the spleen. So that means we are not just killing the cells. So this is an interesting candidate that we are further following up. Another candidate is LRRC8C, which has a similar expression pattern. It's highly enriched in T cells, but not so much in other immune cells. And LRRC8C is one of 5 members of a small family of chloride channels that have 4 transmembrane domains, and that were recently structurally resolved by Raimund Dutzler's lab in Switzerland. His lab has shown that LRRC8A, the most important member of this family, forms a hexameric structure with a large pore. But we are mostly interested in LRRC8C because it's so highly expressed in T cells. This is again shown here in mouse and in human T cells. These pie charts basically just, or donut charts, basically just show that it's really 8C and not compared to the others that's highly expressed in T cells. So when we delete LRRC8C in knockout mice and then take the T cells from these mice, we find that they do not, that we have abolished currents, so-called volume-regulated anion currents, REC currents. And these are currents that are induced by hypotonic cell swelling. When you put cells in a hypotonic buffer, they become larger because they're taking up water and they swell. And this induces the opening of the REC channels. But if you knock out 8C and T cells, you can basically prevent or can completely abolish these currents. And so what these channels are important for is what's called the so-called regulatory volume decrease. Basically, after cell swelling, osmotic cell swelling, the cells become larger, and this leads to the opening of RAC channels. Chloride and organic osmolytes are extruded, and this leads to a passive efflux of water, and the cells start to shrink again. And one can basically visualize this with a dye. In wild-type cells, after osmotic challenge, the cells swell, and then they shrink again. Whereas knockout T cells from LRRC8C knockout mice also swell even more, but they shrink less. Their rate of cell size reduction is less, and it's quantified here. So that means that 8C is important for the regulatory volume decrease. Now, the problem for us was that we couldn't really think of a scenario in which T cells really recognize hypotonic conditions. And so this puzzled us for a long time because we didn't know what this channel is really important for. But when we stimulated T cells through the T cell receptor and then looked at proliferation and cell death in response to T cell activation, we found that in the absence of LRRC8C, T cells can proliferate more, indicated here by the red peaks. And the reason for that is that more of these cells are in the G2M phase of the cell cycle, which is again quantified here. At the same time, the LRRC8C knockout T cells also die less. They have undergo less apoptosis after stimulation, shown here by Annexin 5 and caspase 1 staining. So we have more proliferation and less cell death. And we didn't know for a while what caused this defect. And so we did an RNA-seq analysis of these knockout T cells. And the most prominently deregulated pathway we found in several analyses was the P53 Sickling Pathway. And this is obviously a very important pathway in cancer cells because it imposes cell cycle arrest and apoptosis on cells normally. But when mutated, when P53 is mutated, this can lead to cell cycle progression, and which promotes tumor growth. And without going into many experimental details, I just will tell you that upstream of P53 is LRRC8C. And the way it works, we think, and we showed this in multiple experiments, is that it mediates the uptake of a second messenger that's called 2,3-cGAMP, which turns out to be an important immune signaling molecule. And there were two papers last year by a group at Stanford and one in China that showed that in epithelial cells and macrophages, LRRC8A, together with other subunits, can mediate cGAMP uptake into cells to activate a molecule called STING, and thereby to activate the interferon alpha and beta immune response. What we found is that in T cells, LRRC8C through cGAMP and STING regulates P53 activity. And we can show this basically by treating cells with 2,3-cGAMP and then looking at proliferation and CD80 expression, which is downstream of P53, and see that this is strongly disturbed in the absence of LRRC8C. So this seems to be an important pathway in T cells that regulates T cell proliferation and cell death. Now, the second approach that we're using to identify new ion channels is done by 4 genetic screens, shRNA screens, and now replaced by CRISPR screens. And the general screen principle is basically shown here. So we are taking T cells from T cell receptor transgenic mice that recognize either virus or tumors or something else. And then we take these T cells, transduce them with our pooled SH or CRISPR libraries, expand these cells briefly in vitro, and then we re-inject them into mice. And these are typically mice that don't have T cells of their own, so that all T cells in these mice are of donor origin. And then we challenge the mice with a virus infection, autoantigen, tumors, and so forth. Then we wait for several weeks for the in vivo selection to take place, isolate T cells again from the mice, and then at the end of this process, we are doing next generation sequencing to see which T cells have been depleted because of their knockout of certain ion channels. And we have done over the last years several of these screens in vivo to test the role of ion channels in various T cell subsets. One is to study the role of ion channels in CD4 T cell expansion in response to virus infection, either LCMV or influenza in vivo. We have tested the role of ion channels in anti-tumor immunity in CD8 T cells by looking at CD8 T cells that infiltrate tumors. And we have also used 2 models of autoimmunity to test the role of ion channels in Th17 cells in the context of EAE and inflammatory bowel disease. We are also doing tests or screens in vitro, one for the role of ion channels in macrophages, so-called myeloid-derived suppressor cells that regulate anti-tumor immune responses, and another screen in B cells. Because in B cells in particular, the role of ion channels is not well studied, but B cells are important to make antibodies, for instance, in response to infection. And I want to give you 2 examples. One is the screen in B cells, and this is a project driven by an MD PhD student in my lab, Tony Tao. So B cells undergo several rounds of what's called affinity maturation in a structure called the germinal center in lymph nodes and in the spleen. And in this process, the B cells will refine their antibody, the affinity of their antibody, and also undergo a process that's called class switching, or CSR, class switch recombination. And what this means is that they switch from predominantly IgM producing cells to IgG, IgA, or IgE producing cells. These are antibody subclasses with higher affinity. At some point, B cells can leave the germinal center and become long-lived plasma cells. And these are really antibody-producing machines that produce large quantities of antibodies that we need for immune responses. So in one screen, Tony has tested the effects of ion channel deletion on class switch recombination and also on long-lived plasma cells. And for that, he's using an in vitro system using B cells isolated from Cas9 transgenic mice that are transduced with a library of small guide RNAs, 1200 ICTs and metabolic transporters. In the first step of the analysis, you can look at class switch recombination, B cells that produce IgM and then switch to IgG1. And in the second step, we can look at plasma cell differentiation and look at B cells that express CD138. And by then doing next generation sequencing, he can basically look at the depletion of ion channels from the class switch B cells or the plasma cells. And reassuringly, he finds a couple of positive control genes like AID or BLIMP1 that are required for these processes, but he also finds ion channels and transporters circled in red here that may influence these processes. And he is currently in the process of following up these candidate genes. Now, the second example I want to show you is an in vivo screen conducted by Liwei Wang in my lab. He's a postdoc. And he is using, again, the EAE model of multiple sclerosis. And it's the same or very similar setup that I mentioned before. He's starting with CD4 T cells that specifically recognize myelin. These are from so-called 2D2 mice. He's transducing them with an shRNA library. And then he's basically re-injecting the transduced cells into mice, induces EAE. And after several weeks of in vivo selection, he's isolating cells, T cells, donor T cells from the spleen and the CNS. And then he looks for the depletion or enrichment of candidate genes. And this is a representative Z-score plot in which we basically plot the depletion of ion channels and transporters in the spleen versus enrichment. And on the y-axis, we are plotting enrichment and depletion of ion channels in the CNS. And this is really what interests us, the genes that are falling into this pink box here, because these ion channels, deletion of these ion channels would lead to depletion of T cells, specifically in the CNS. We are also interested in these ion channels that lead to depletion in the CNS and in the spleen. And he identified overall 43 candidates that fall in different clusters. And I'm showing you examples of HIT6 and HIT6 and 7, which are not very highly expressed in T cells that we isolated from the brain and analyzed by RNA-seq analysis. But when we delete these ion channels by CRISPR-mediated gene editing, and then inject these T cells back into mice, you can see that we basically prevent the development of EAE in these mice compared to the controls. And the reason for that is that the T cells do not accumulate in the brain, into the CNS of these mice compared to the controls, but they are present in the spleen, actually in larger numbers compared to the control. This means we are not just killing the cells by transducing them with the ion channels or transporters, but we're basically selectively preventing their accumulation in the brain, either through differentiation, proliferation, or migration to the brain. So overall, we are doing all these screens that I mentioned before. And the idea behind this is that we hope that at the end of this process, we can identify ion channels that selectively regulate the function of macrophages, B cells, CD4 and CD8 T cells, or Th17 cells in the context of different disease processes or biological functions. And the hope is that we can potentially modulate these ion channels, for instance, in anti-tumor immune responses without affecting antiviral immunity or antibody production by B cells. With that, I end this part of the talk. I just want to thank the people who have been involved in these studies. And I mentioned Axel, Liwei, and Tony, but there are also other people like Tiger and Lucille, who are doing screens on these projects now. If I have 2 minutes, I just want to briefly mention that in order to support this research, I have several years ago started a program that's called the Ion Channels and Transporters Program in Immunity, ICTI at NYU. And this is supported by the School of Medicine at NYU and the Department of Pathology and the Office of Science and Research. And this program really consists of several pillars. One is interdepartmental collaborations and work in progress seminars. We have seed funding. We have an Ion Lab core facility that provides electrophysiology services. We have an education component. We started a new Ion Channel graduate course in which Tony and Axel also help to teach. We have a seminar series, and we are also organizing conferences and symposia. And there's one symposium that in particular, or conference, that I want to draw your attention to, and this is the 74th annual meeting of the Society of General Physiologists in September this year. We had to postpone it last year because of COVID, but we hope to have an in-person meeting later this fall. And the topic of the meeting is ion channels and transporters in immunity. And so we have 3 full days of sessions that are all dedicated to this topic. And this, to my knowledge, is really the first conference on this topic. So I hope some of you may be interested to join us for this. And because it's an unusual combination of topics, immunology and ion channels, we decided we will have an educational workshop to start off this conference on May 21st. This is fully online. And we were fortunate to get a couple of real leaders in the fields of immunology and ion channel structure and function, including Lily Jahn, Stephen Long, Hida Plu, Rich Luce, and Leslie Burke, to give an introduction to their topics to researchers on the other side. So it's basically, we call it ion channels for immunologists and immunology for physiologists. And you're all welcome to attend this meeting. And I think this will be very useful to bridge the gap between these two fields. And with that, I thank you for your attention, and I'm happy to take any questions if you have some. Thank you.
Great. Thank you very much, Stefan. We have one question in the chat box from Kevin.
Hi, Kevin. What activates LRC8 in T cells? Yeah, that's a good question. So as I mentioned, we were puzzled by this, right? Is it really a hypertonic environment? Typically, what people use to activate Rag currents in in cells is a very hypertonic solution, and we couldn't really come up with a scenario in which this may happen. So what we do find is that when you activate T cells, they will commit apoptosis. So they will die, or they will undergo other forms of cell death. And that may lead to the fragmentation of DNA or a release of mitochondrial DNA that then stimulates this innate, typically this innate immune sickling pathway, the cGAS enzyme that generates cGAM. And cGAS and STING are also expressed in T cells. And what we think is that the cGAM produced by cGAS is actually released by T cells through LRR channels or other transporters. There have been a couple of others shown to also mediate cGAM transport. And this in a paracrine or endocrine, paracrine or autocrine manner can then stimulate T cells, activate T cells. So we think that this response in immune cells or in T cells has not much to do, if anything, with hypertonic conditions. I hope that makes sense.
Great, thanks. And you have another question from Mark Haynes.
Mark Haynes, are there specific ion channels identified that affect migration-specific events such as integrin expression activation? For instance, in one context, B cell development depends on TFH cell migration into lymph nodes. Yeah, that's a great question. So we could either start base a screen on these properties to look which ion channels regulate migration, and then that migration would be our readout. We have not done that. We are tackling the problem from the other side, right? We are looking at a complex in vivo scenario, and then we are tracing our steps back. And you're right, so we can either look for, especially in the context of EAE, right? We don't have T cells in the brain. So this could also be a migration defect for TFH cells or B cells in the lymph nodes, this could absolutely be a migration defect too. Although I will say that for our screens in vitro, this may not play such a big role, but if we had done this in an in vivo screen, for sure. We have not identified ion channels that specifically lead to migration, but we have just not looked at this very carefully yet. But this will come.
Great. Thank you very much. Great work, Stefan, and fantastic work, Niranj. Thank you so much for attending and presenting and sharing your work today. Again, I'll encourage everyone to contact the speakers if they have any questions. I'm sure they would be very happy to answer you by email. I'll also remind the participants that we have that this IDG e-Symposium series, the next one's going to occur on May 4th. Anna, can you give any more details on this? Oh, fantastic. So the next theme is going to have to do with informatics tools. We've got Paul Sternberg and Lincoln Stein as speakers, and I'm sure we'll have a really fantastic data to share. Is there anything else that I should add, Anna, before we close the meeting?
Nothing from my side. I just want to say thank you to both of the speakers for participating today and to all the attendees. And we are so happy that you're showing interest in IDG, and we hope you continue.
Wonderful. Thank you, Anna. Thank you again, Stefan and Niranj.